in the last few lectures, I have first taught you about that about the definition of biomaterials, biocompatibility, host response, and as well as the importance of biomaterials. Then that was followed by the molecular biological description of biological cell. So, molecular biological description of biological cell, that time I have focused on that, definition of the cell, what are the different components or the cellular organelles, and how their structure looks like in terms of the membrane structure, like it has a double membrane structure. Most of the cellular organelles have the double layer at the membrane, and they are enclosed by the double layer membrane. And also on the cytoskeleton, like what are the different components of the cytoskeleton, like actin filaments, microtubules and intermediate filaments, how their property looks like. In today's lecture, I will continue the description of the further molecular biological aspect of the biological cells in terms of the cell cycle, and some other relevant information which should be useful in understanding the cell material interaction as well as the cytocompatibility or biocompatibility behavior of the materials. So, cell cycle. Now, one biological cell, as I said in the definition of the biological cell, that a cell can be defined as a self duplicating unit capable of duplicating itself by genetic information which is enclosed in its nucleus. And therefore, that when you start with that any particular biological cell, that chromosome duplications and cellular growth that occurs. Now, cellular growth is a part of the cell fate processes. Cell fate processes means it is a combination of cell proliferation, cell adhesion, cell differentiation, etc. Now, when the cellular growth takes place, then cell also increases in size, because I will mention it to you later on that cell has a kind of very much elastic properties, and cell can undergo different cellular adaptation process depending on the environment as well as external stimulus, etc. Now, if you see here, that is a chromosome segregation takes place prior to the cell division process. And cell division also composed of two steps. In the first one is the cytokinesis, and then second one is mutation. And cytokinesis, you can see that this chromosome segregation takes place, and then cells are also going through a transition in terms of the cell shape. You can see the cleavage in the cell shape here, and you can see this kind of cleavage. So, that is kind of onset of the cell division process, and subsequently the one given cell, it can lead to the formation of the two daughter cells, and these are the two daughter cells, and these two daughter cells can undergo again chromosome replication in the cell growth processes. So, this is the way cells multiply in a given biological environment. Now, cell death, it is also known as necrosis or also programmed cell death. Now, when cell death takes place, typically you can immediately realize on the scanning electron micrograph or in the fluorescence micrograph, if the cells appear like in a typical globular shape or typical spherical shape, then you can very well say that cells undergo apoptotic processes. Cell apoptosis means cellular death. Now, you can see that this is the kind of very much spherical type of cells which are typically observed in the case of mammalian cell lines. Now, how the changes in cell shape processes take place? As I said, that cells can change its size, cells can change its shape depending on the external environment. Now, if you see the first step, like you have small soluble subunits like protein molecules, and then it can form a large polymers, filamental polymers in the cytoskeleton, and if they get a signal such as a nutrient source or something, then that acts as a signal, and then you can see that cell also changes its shape. It is a nucleus, these are like globular soluble subunits, and these are like filamental forms, which is nothing but protein aggregates. Now, what they do, that this large filamentous polymer, they can also break down to smaller globular subunits, and subsequently they can reassemble by taking the proteins or signals, taking the nutrient source from the external environment. So, entire process leads to the increase in the cell size as well as the shell shape. So, that is why it has been written here, regulated filament assembly of small proteins and rapid reorganization of the cytoskeleton. So, essentially you have to remember all the time that shell shape process is nothing but reorganization of cytoskeleton. Cytoskeleton is kind of a reorganization of cytoskeleton, and this cytoskeleton composed of actin filaments as well as the intermediate filaments and so on, and microtubules. So, if they can either disassemble or reorganize itself when they receive the external stimuli, then cells can change its shape. Now, this is the typical structure of the, because till now I was showing through all the different structure of the cellular organelles. Now, this is the typical structure of the platelets, blood platelets. It is like one of the entire structure. You can see this is like a half of the structure, essentially to show you the internal organelles which are present there. Now, as you can see, these are like globular subunits which is known as the glycogen. You have also secondary vesicles here, and you have the mitochondria. Mitochondria, as you know, it also is enclosed by a double membrane, and these platelets are circulated in the blood and they stimulate blood clotting at the sites of tissue damage and thereby preventing excessive bleeding. So, once the blood clotting takes place, then bleeding can be stopped, and that is essentially enhanced by the presence of the platelets. Now, couple of slides back I explained to you the structure and composition of the human bone, human natural cortical bone, and there I said that cortical bone can be described as a polymer ceramic composite. You have the 65% collagen and you have the 65% hydroxyapatite particles, nano-sized natural hydroxyapatite particles, and you have also 25% collagen. Now, collagen is essentially polymer, hydroxyapatite is essentially ceramic, and therefore, this bone can be very well described as a polymer ceramic composite. Now, structure of collagen, if you look at the various level, now it typically it has a very micro and ultra micro scale. It has a triple helix type of structure. Triple helix means you can see that this is the one part of the collagen, this is the dotted is the another part, and then third one is that typically collagen composed of triple helix structure, and then entire microfibril, it is somewhere around 300 nanometer. So, these kind of numbers are important, so that you understand that that collagen, what is the typical length scale, and I mean how collagen also looks like. So, further you can see that how this collagen fiber takes place. At the micro scale, I say that collagen microfibril, and this is that one of the microfibril. So, expanded version or the magnified version is this one, which has a typical length of up to 300 nanometer. Now, these microfibrils essentially they aggregate first, and then these cluster of microfibrils, they are bonded by the hydrogen bonds, by lateral hydrogen bond, and that leads to the lateral aggregation, and this end to end aggregation that leads to kind of a collagen fiber. You can see it is like a fiber bundle. If you remember in the material science, many times people teach you about the fiber reinforced composites. So, how the fiber looks like, so that is how the collagen fiber also will look like, and but this collagen fiber basic building unit is a triple helix structure which has a length scale of the around 300 nanometer, and on an individual triple helix it has a length scale of 86 Armstrong, which is extremely fine, and this is that glycoprotein and then other type of proteins. And collagen, there are several types of collagen like collagen type 1 and so on, that depends on the how the structure of the collagen, individual collagen microfibril, they changes in terms of composition as well as orientation. This is like more details about the collagen fiber structure. You can see that this is one collagen microfibril aggregate, this is another collagen microfibril aggregate, this is the third aggregate. So, this is just part of the collagen, large collagen fiber, and each microfibril aggregate, if you notice, they have a kind of dimension of around 4 micrometer. 4 micrometer, that is the kind of a diameter, and unit fibrils they have a radius of 800 to 1000 Armstrong. So, this is the typical dimension of the collagen fibers structure. Now, how this compact bone looks like? Now, you can very well realize that how does this compact bone they look like in real life. Now, in the compact bone you have this collagen fiber matrix, and then you have these very nano sized hydroxyapatite particles which are dispersed uniformly in the matrix. And if you look at this little bit in details of this how this compact bone looks like. Now, first one you can see that Haversian canal. And what is Haversian canal? It is a kind of a channel which supplies the nutrients and the blood to the bone side. Okay. Then second one is the cement line. This cement line is a kind of boundary of osteon, and osteon is nothing but this is like individual osteon here which is shown, and this is like osteon lacunae. Now, cement is required to build a house, right, because what it does, so it essentially makes the bond between the two bricks. So, similarly here the cement line means it is a biological cements which actually acts as a glue to put all the structure collagen hydroxyapatite structure together. And this is the structure of the cancellous bone. So, there are two types of bone as I mentioned, one is the compact bone, one is the cancellous bone. Cancellous bone is more porous type of structure, compact bone is more strong type of structure because it is a non-porous type of structure. And in the compact cancellous bone you can see the connective tissue, and that cell lines of the connective tissue, as I said, fibroblast type of cell lines. You can see that osteons, which is the very small subunit of the typical contact bone. You can see that osteons and Haversian systems, that is the Haversian canals and so on. And you can also see the periosteum. Periosteum means like you have like a shirt, in a shirt has this kind of cover, right. So, like similar things it is also present in the bone, and this periosteum region is that outer part of the bone structure. So, that is what is known as the periosteum. This is the unique three-dimensional structure of the cancellous bone. What you can see that it has a highly porous structure, and this porous structure essentially leads to very weaker mechanical properties of the cancellous bone. Whenever a structure is porous, then it has a poor mechanical property, whereas compact bone, which is a very non-porous type of structure, very strong structure, it has also very good mechanical properties. This is the typical structure of the tendons, and these tendon cells typically present in a kind of a row, and mostly it is a fibroblast or connective tissue cell lines which are present as an aggregate in a number of cells which are kind of organize themselves in a particular fashion, and you also have the collagen fibers in these tendon cells. Now, from the typical cell size and number of molecules, you can find out that what is the typical clinically relevant number of cell lines. Let us say these 3T3 cells, this is a fibroblast type of cells, like you can use that L929, that is mouse fibroblast cells. You can also use these 3T3 cells, this is also again fibroblast type of cells, and it has a typical diameter of 15 micron. So, 15 micron diameter means you can calculate the volume as 2 into 10 to the minus 9 centimeter cube. And if you consider the bacteria, which is a 2 micron in length and 0.8 micron in diameter, now it has a volume of typically 1 into 10 to the power minus 12. This is like centimeter cube. Now, what you can see here, the typical bacteria like Escherichia coli, which is a long shape, and this long shape means it has a particular aspect ratio, like it has a particular length and particular diameter. Now, if you typically take this length and diameter as 2 micron and 0.8 micron respectively, then you can calculate the volume is 1 10 to the minus 12 centimeter cube. If you consider that versus the volume of a cell, now volume of a cell is clearly 3 orders of magnitude larger compared to that of the bacterial cell lines. Then protein concentration in cytoplasm is typically 180 milligram per milliliter, and average protein mass is 50 kilodalton, and therefore 3.2 million proteins or 2 into 10 to the power 8 molecules per milliliter of proteins is available in the cytoplasm, and therefore number of proteins per cell is 4 into 10 to the minus 4 into 10 to the power 9 molecules per eukaryotic cells or 2 into 10 to the power 6 molecules per prokaryotic cells. So, eukaryotic cells means cells with nucleus, prokaryotic cells means cells without nucleus, that is the example of the bacteria cells. Now, what you can see from the simple calculations that you can have 10 to the power 9 molecules per eukaryotic cells like mammalian cells or biological cells, whereas you can have 10 to the power 6 molecules because there is a 3 orders of magnitude difference in terms of the volume. So, 10 to the power 6 molecules in the case of the bacterial cells. So, it is a huge number of protein molecules, they are enclosed within a single unit cell, because from the engineering aspect it is important for you to have an idea of the numbers. Like when you say that large number of protein molecules, people would immediately ask you what is the typical number. Typical number is 10 to the power 9 order of magnitude protein molecules, they are contained in a single biological cell. 10 to the power 6 number of molecules, they are contained in a single bacterial cell line. Now, bone composition wise, bone has an organic framework. Organic framework means it has a kind of a it is mostly like like either cells or collagens or other organic molecules. Now, cells, it can have bone cells, osteoblasts, that is the bone formation cells. It can have also osteocytes, and you can have osteoclasts. Osteoclast is the bone resorption cells. Bone resorption cells means if any bone they have a large number of osteoclasts, then this bone will dissolve in the in vivo environment. So, bone cannot survive, but if you have large number of osteoblast cells, that means bone growth can take place, that bones can survive and bones can grow. Now, you can have collagen also in the bone, as I said, and other organic molecules. Now, inorganic salts is that calcium and phosphorus. So, calcium phosphate type of crystals, they are present in the major fractions, and therefore, bone stores minerals and ions for body functions. This is one of the important factor why we always test as osteocalcin or the bone mineralization test as part of the in vitro study. For any synthetic material, you would like to check whether it has a bone mineralization ability or bone mineralization property for that particular material. And osteocalcin actually tells you that whether this bone mineralization is capability of a particular material. Now, cell injury, cells actively control the composition of their immediate environment and intracellular milieu. Intracellular milieu means within a cell membrane, the area which is enclosed by the cell membrane, within a narrow range of physiological parameters like homeostasis. So, physiological parameters means like pH, like temperature, etc. So, essentially what is the meaning of the first statement, that cells will actively control the composition of their intermediate environment all the time, and they also try to maintain the environment of the intracellular space. And under physiological stresses or pathological stimuli, injury, so physiological stresses or pathological stimuli like you are getting some wound or you are having a accident or fracture, then under this kind of conditions cells can undergo adaptation to achieve a new steady state that would be compatible with their viability in the new environment. What does it mean? This means when you are getting some damage, physical damage or physical injury, then cells will quickly adapt so that cells can survive under the changed environment, under the changed scenario. So, that is a unique capability of the biological cells, that cells can change its size, shape or whatever physiological conditions, the way external environment also, I mean with the change in the external environment. So, like in the normal conditions, normal healthy conditions, cells will have one set of physiological condition or physiological situations. When the external environment changes, cells can change itself so that they can survive well. Okay. So, that is the unique capability of biological cell. If the injury is too severe, irreversible injury, then affected cells die. Now, however, this adaptation, cellular adaptation to the new environment also can take place to a maximum extent. Now, if the injury is too much, then cells will die, and that is what takes place in the case of paralysis or any other kind of disease. Then what happens, that tissue or organs that completely fail to do its original function. For example, paralysis means hand paralysis. So that hands cannot, you cannot really do any movement, or leg side paralysis, that means legs cannot move itself. So all the tissues and the cells, they cannot perform their normal function because the injury is too severe, right. So that is the meaning of these particular statements in this slide. Now causes of cell injury. Now there are different kinds of causes. So first one is the hypoxia and ischemia. Hypoxia means lack of oxygen. So when there is a lack of oxygen at a particular implant site, then what will happen? It will have a kind of hypoxia situations, like that these cells will essentially lose their normal function. Then chemical agents means like some particular chemical agents, they enter the body immune system. Infections, several kind of infections is possible. Immunological reactions, genetic defects. Genetic defect means inside the nucleus at the DNA RNA level, if you have any genetic defects. Nutritional defects and aging. Aging means like when a people become old, they cannot walk that fast. Why? Because the normal function of the cells, that also deteriorate with aging. Like the more you become older, the less you become more and less you become active. So that is the reason that aging also causes the cell injury. It is not a cell injury, it is like that functioning of the normal cells will not be there. That many times, that is why people stress upon in biological experiments that it is a healthy cell line. Healthy cell line means cells are at their optimum stage of functioning. Okay. So that is called healthy cell lines. Hypoxia and ischemia. Hypoxia is a state of oxygen deficiency in the body which is sufficient to cause an impairment of function. So it's oxygen deficiency that causes an impairment of function, and it is caused by inadequate supply of oxygen transport or the inability of tissues to use oxygen. So there are two things it can happen. One is that there is an inadequate supply of oxygen, that means you do not have sufficient amount of oxygen available or access around the cell, or the other things is that tissue, although oxygen is available, but tissue cannot actually use, or tissues are not able to use that oxygen to do its normal function. Now ischemia is another kind of cell injury. It is a lack of blood flow and oxygen to the heart muscle. So this is a small spelling mistake, heart muscle. So what it means? It means that it is lack of blood flow, and through blood your oxygen also is circulated. So if there is a lack of blood flow to a particular location, then what will happen? Your tissue or cells, they will not be able to get sufficient amount of oxygen, and that is caused by the restriction of the blood flow to a part of the body. Now cellular adaptation, as I said, can be induced or regulated at any of a number of regulatory steps, and these steps includes receptor binding, signal transduction, gene transcription, or protein synthesis. So the cellular adaptation process can be regulated at any of a number of regulatory steps. And what are the 4 different adaptive changes that cells can undergo? First one is atrophy. Atrophy means decrease in the cell size, like cells will get squeezed. Then hypertrophy, that is increase in the cell size. Hyper means more, hypertrophy means it is an increase in the cell size. Atrophy means it is less in cell size, that is decrease in cell size. Hyperplasia, that means increase in the cell numbers, and metaplasia, that is in change in the cell type. Now let me give you some examples. For example, metaplasia, if osteoblast cells, they transform to, or they go transition to osteoclast, then it is an example of the metaplasia. You remember, osteoblast is the bone forming cells, osteoclast is the bone resorption cells. So if the bone forming cells by certain biological process, if they undergo transition to osteoclast cell lines, then it is this process is called metaplasia. Hyperplasia means it is a increase in the cell number. That means under certain changes in the external environment, cells are undergoing more number of cell division process, so that you know that number of particular cell type actually increases. Now those kind of situation or cellular adaptation process is known as hyperplasia. Remember this 4 cellular adaptation process, that is cells undergo when there is a change in the external environment of the cell. Little bit more description of this different cellular adaptation process. Now atrophy. So atrophy is the shrinkage of the cells, and how this shrinkage takes place. Now physically you can see that if there is any change in the size, that size change or decrease in the size can only be accomplished when the cellular content is somehow squeezed out, right, and that is a loss of the cellular substance, and with the involvement of a sufficient number of cells, an entire organ can become atrophic. Now what is the genesis of these things? Now as I explained to you earlier, cells they will form tissues. Tissues is like what? Tissues are defined as a self-organized structure of a number of cells which are organized in a particular fashion to form a tissue. Now a number of tissues they form a organ. Now what is explained here, that if a number of cell lines or sufficient number of cells are becoming atrophic, means like they really decrease in size, so the entire organ which is composed of that particular cell line can become also atrophic in nature. And these causes of atrophy include decreased workload, like you are very lazy, you do not work, you always sleep and sit, and then you have atrophic, then decreased pressure, diminished blood supply or nutrition. So these are like atrophic nature, and also aging. Hypertrophy is again is increase in the cell size, so that is possible by gain of the cellular substance. And now with the involvement of a sufficient number of cells, an entire organ can be hypertrophic, just like atrophic, entire organ can be atrophic. Hypertrophy is caused by increased functional demand or by specific endochronic stimulation. Not only the size but also the phenotype of individual cells can be altered in hypertrophy, and with increasing demand, hypertrophy can reach a limit beyond which degenerative changes and organ failure can occur. Now what is the last statement that tells? That tells that hypertrophy, that if the cells increase their size and therefore a number of cells they contained in organ that also changes its size, then it undergoes degenerative changes. Degenerative changes means normal biological function cannot take place, and an organ failure can essentially occur. So organ failure means the people still can have that particular organ, but that organ cannot work or that organ cannot function in a normal sequence. Hyperplasia, that is constitutes an increase in the number of indigenous cells in an organ tissue, and pathological hyperplasia occurs if typically the result of the excessive endocrine stimulation. Now metaplasia is a reversible change in which one adult cell type is replaced by another adult cell type. That is the example that I have given to you, that is the osteoblast cell lines, they undergo or they change it to osteoclast. And metaplasia is a cellular adaptation in which the indigenous cells are replaced by cells that are better suited to tolerate a specific abnormal environment. Abnormal environment means when the external environment is not suitable for the survival of that particular cell lines, then they quickly change to another cell type, and the example I have given for the bone cells. So here you can see that typical bone structure in terms of the different cells they are present in this bone. So osteoblast cells, they have a typical irregular type of cells and with a nucleus. You also have osteocytes, which has also typical star type of cells, and the star type of cells, star shaped cells, it has also central nucleus. So these are examples of the eukaryotic cells. Now these are also, these osteoblast cells are of different sizes here, and you can see Golgi apparatus, mitochondria, all these important cellular organelles. You have the calcified bone matrix. Calcified means it has the hydroxyapatite, that is why it is called calcified. So whenever you see that calcified bone matrix, that is like many times when you visit the doctor, doctors will tell you to take calcium tablet. Why calcium tablet? Because your bone has loss of calcium. So, to compensate with the loss of calcium, you need to take the calcium tablets, so that certain calcium level is maintained in your body. So, that is the scientific meaning of taking these calcium drugs. Now, ECM, as I said earlier, ECM stands for extracellular matrix, and extracellular matrix contains typically collagen. It has also fibers like elastin fibers. Remember, I have mentioned that this extracellular matrix contains different fibers, and that fibers give the ECM mechanical support. So, ECM you can consider as a matrix where the different cells are present. So, that ECM has to support support all the cells that it contains. So, if it has a fibers like structure, then it is mechanically also suitable. You have the different proteins which is called fibronectin, and also it has integrins. Now, what is the biological roles of the extracellular matrix? Like as I just mentioned to you, that it acts like a mechanical support. It also determines the cellular polarity. Many times cells have electrical charges, and it has positive and negative polarity, that is also controlled by ECM. It also controls the cellular growth or cell fate processes, control or maintenance of cell differentiation. Now, cell growth, cell proliferation, cell differentiation, these are like part of the cell fate processes, and scaffolding for tissue removal, establishment of tissue microenvironment, storage and preservation of regulatory proteins. So, these are like all the different biological roles of the extracellular matrix. Now, elastin fibers, as I mentioned to you earlier, that extracellular matrix, they have this collagen fibers, they have the elastin fibers, they have fibronectin protein, they have laminin. Now, the question is that how the elastin fibers they look like. Now, elastin fibers, if you see that the shape is more like a snake, like you have it is squeezed or something, and these are like protein based fibers. However, there is a link between these two fibers here, and this link is weak link, and this weak bonds is mostly hydrogen bonds. Now, when you stretch it, what is shown here, that when you stretch it like tension, then what will happen, this elastin, that there is a cross linking between these two fibers. So, it is like you can say polymer macromolecular structure, each chain, two neighboring chains, they have a cross link, and typically in polymers this cross linking is taking place by adding sulfur to the polymer, like. So, due to the presence of certain elements, this cross linking is established. Now, when this squeezed kind of shape it is stretched, then you can see that all these fibers, they are also equally stretched in a particular orientation, that is the orientation along the tensile axis, and two fibers, their edge bonds or their cross linking bonds, these are clearly visible, but it has a highly elastic nature. If you release the load, then it can go back to the original coiled kind of configuration. So, this is like what we call coil type of configuration, and this is more like a more expanded version, and this expanded version is more like a that individual elastin fibers, they are getting stretched, and therefore, you can see like more like when the fibers are getting stretched, how they look like. The similar conditions you can find it here. This is more kind of vivid or more detailed description how the collagen fibrils they look like. Now, if you see this collagen fibril, they have like different numbers are there, and these numbers are like you have a 280 nanometer. So, this entire length is kind of 560 nanometer, this entire length, and they have a triple helix type of structure, right, and individual collagen bundle, they contains that number of collagen microfibril aggregate that I have shown you earlier, and this each microfibrils, in each of the microfibril aggregate, individual microfibril, they are attached to each other by the weak hydrogen bonding. Okay. Coming back to the description of the collagen fibrils, so what you can see here, that it has this long microfibril type of structure which are attached to each other by hydrogen bonding. They form this microfibril aggregate which are contained in a large collagen bundle or collagen fiber. Now, mechanisms of to cell injury, like the cellular adaptation or cell response to injury is not an all or nothing kind of phenomena, and cells are complex interconnected systems, and single local injuries can result in multiple secondary or tertiary effects. So, secondary or tertiary effects means cells can change shape or change or type. So, shape change is called atrophy or hypertrophy, number change is called hyperplasia, type change is called metaplasia, and cell functions is lost far before biochemical and subsequently morphological manifestations of injury becomes detectable. What it says that cellular that organ failure, like when a human patient has a failure of the organ, that takes place much before that this is detectable, because all these biochemical and morphological manifestations that takes place first, leading to the final organ failure. So, that needs to be understood. And what are the different biochemical mechanisms? That is, number one is the loss of energy, like ATP depletion or oxygen depletion. ATP is that adenosine triphosphate. Mitochondrial damage. Mitochondrial damage, if any damage takes place in the mitochondria, mitochondria is known as the energy store of the cells, like it is the place where the energy is produced by the reaction of the ADP to ATP conversion, right, and that conversion adds energy. Now, if the mitochondria itself is damaged, that means cells will not be able to produce energy for its own survival, and if the cells do not have sufficient amount of energy, cells cannot survive by itself. Because remember, the basic definition of the cells, it is a self-contained unit, like whatever is required for its own survival and duplication, that is already contained in the inside the cell. Like mitochondria is energy storage, you have the protein synthesis unit, that is itself is required for its own survival as the extracellular matrix, that is the ribosomes. You have that genetic transformation or this genetic translation, like cell division, that is contained in its nucleus. So, all those units are contained in the cell. Then third one is the loss of calcium homeostasis. So, as I said that bone contains that collagen as well as the synthetic natural hydroxyapatite, and if the calcium deficiency is there, then also there will be bone loss. Then defects in the plasma membrane permeability, and fifth is the generation of the reactive oxygen species O2 minus, H2O2 or OH minus and other free radicals. Now, if there are other free radicals and oxygen species are present, then also cells can die, because it can directly attack the cellular membrane, and if the cellular membrane is damaged, then that all the internal intracellular milieu whatever is there, that can come out. So, cells cannot survive like. Now, what is the cell numbers in tissue biology, kind of orders of magnitude? Now, in the in vivo conditions, the whole body, it has a in vivo condition is that 10 to the power 14, and human organ is 10 to the power 8 to 10 to the power 11. Now, what is the cell production in vivo? Maximum from a single cell, that is the 2 to the power 30 or 2 to the power 50. That is the huge number of cells can be produced, which is roughly with 2 to the 30 to the 50, a huge number of cells that can be produced from the individual single cells in the in vivo conditions. Remember, in vivo environment and in vitro environment is different, and accordingly this number of huge number of cells may not be generated in the in vitro conditions, right. So, this is under in vivo. Now, cell production ex vivo, like requirements typical cellular therapy. Ex vivo means outside the patient or outside the animal. So, that is 10 to the power 7 to 10 to the power 9 number of cells, that is a large number of cells there can be produced in the ex vivo conditions. Now, expansion potential of human tissues in vitro, like there are different CD34 enriched or 2 to 3 antigen enrichment, like they can be expanded like many orders of magnitude, like dermal fibroblast, that can be more than 10 to the power 6 fold increase or expansion that is possible in the in vitro conditions. Now, a typical organ which contains 100 to 50 or 500 milliliter in size contains 100 to 500 billion, that is 10 to the power 9 number of cells. So, a typical organ contains huge number of cells, that is 10 to the power 9, and this organ contains functional subunits which is like each of the linear dimension of 100 microns, and the cell number in a cube of 100 micron size is roughly 500 to 1000. So, each cube, individual cube, that itself contains 500 to 1000 cells, and therefore, a typical organ will have a few hundred million functional subunits. Functional subunits means what? Like in an individual organ, the subunits, they are supposed to perform different functions for that, for that overall, towards the overall functioning of the organ. An individual functional subunit contains that 500 to 1000 number of cells. Now, cell numbers needed for cell therapy protocols, that is between tens of millions to a few billions, like 10 to the 10 to the power 6 to few into 10 to the power 9, and chondrocyte transplants need 110 to the power 6, that is 10 to the power 8, and other examples is the liver support, that requires up to 10 to the power 10 hepatocytes, that is another cell lines, and tissue culture often requires the cells of the 10 to the power 6 cells per milliliter. Therefore, the size of the cell culture device or cell therapies should be somewhere 10 to 500 milliliter in volume. So, typically in your cell culture studies, you often spend how many number of cells, 10 to the 5 number of cells, 10 to the 4 number of cells, that is typically you require, and that is the why they are required, that is explained in this particular slide, that typically you need to disperse this much number of cycles, so that cells will have a good physiological milieu. Now, next few minutes I will just explain to you the typical structure and composition of the biological molecules and the subunits. Now, sugar amino acids, they are like globular or spherical separate subunits, and then they are covalently bonded, then they form the macromolecular type of structure like globular proteins and the RNA, ribonucleic acid. And then if it is non-covalent bonds, that between these two molecules, non-covalent bond, between these two non-covalent bonds, then it is called ribosomes. So, ribosomes has a typically length scale of 30 nanometer. So, 30 nanometer is extremely small, and you can see therefore ribosomes as a small organelles in the cell structure. Now, this is important. Now, protein structure. Now, what is protein structure? At the individual level, protein structure contains one unit of amino acids, and this is the typical composition of the amino acids. You have these organic radicals here R2, and you have this hydrogen in those two ends, and one end you have a nitrogen end, and other end you can have a single bonds, and then again it is connected to another part of this, another MER unit of the protein structure. So, this is a large macromolecular structure, and essentially R1, R2, R3, that can be 3 different organic radicals or the subunits, and depending on this composition of the R1, R2, R3, you can have different type of protein molecules also. So, proteins, as I said, that it have a N terminal amino acids and you have a C terminal amino acids. N terminal means it ends with N, C terminal means it ends with a typical carbon atom. Okay. And you have a both single bond and double bond in this structure, and this is a polypeptide chain that shows the N and C terminal, and it has a covalent bonds across the individual chain. So, individual chain, like different polypeptide units, they are bonded together with the individual chain, and a complete protein structure, protein molecule have both N terminal and C terminal here. Now, this shows more shape or morphology of the protein molecules, like subunits, like a sugar subunits, like hexagonal structure, and then you have the polysaccharides. This polysaccharide structure is composed of this hexagonal structure added to each other by covalent bonds. You have the amino acids, when they are covalently bonded, they form the protein molecules. You have a nucleotide, then when they are covalently bonded, then it is called nucleic acid. And how this formation or this polypeptide chain that take place, that chain formation is possible. Now, you have this particular organic configuration. Now, when it is added with this n number of such MER units, and from this n number of MER units, one H2 molecule is extracted out, and if due to the loss of these water molecules, you get a typical protein structure which has little bit of complex organic formula. Now, amino acids in the peptide bonds, you have this COOH, that is the carboxyl group here you can see, and you have that NH2, that is part of the amino group, and you have also the side chain, that is the side chain is that R1, R2, R3. Remember in the basic protein description that R1, R2, R3 can be different things. Here one of the R is that amino acid, that is that, and that another side chain is the R is that CH3 group, that is the methyl group. Now, as I said, depending on what is the composition of the R1, R2, R3, you can have different protein molecules, that is the fundamentals. You cannot remember all these protein molecule names and their structure, but the fundamental thing is that in a protein molecule you have R1, R2, R3 different organic side groups is possible, and depending on what is the side group, you can have phenyl and alanine, you can have serine, you can have lysine. So, these can have a very complex type of structure of the amino acids. Now, amino linkage and peptide bond formation, what it shows here that you have one amino acid here and you have this one of these MER units here. Now, this H reacts with the OH, and as a result this H2O molecule is taken out from the system. Now, energy from nucleotide and triphosphate hydrolysis, and because of these H2O molecules leaving the systems, you get this yellow shaded region, which is like a peptide bond, essentially peptide bond formation, and a protein molecules therefore forms. An example of the complicated protein molecules, what you can see here, the sequence of the protein molecules, this is also important. Many times one can many times in the literature you can say that protein molecules configuration changes. Configuration changes means like how these protein molecules, how this is your polypeptide backbone. It is just like any organic polymer, it has a backbone structure, and if you have the side group. Now, the side group can be different depending on whether it is a different kind of protein molecule, whether it is a non-polar side chain or whether it is a polar side group, and then orientation of these side chains that determine what kind of protein molecule configuration it will have. So, if the protein molecule configuration essentially means that either you can this position or the orientation of the side groups, how does it change, and how these sequence of these different polar groups they change because of some biological change, because of some condition in the biological changes. Now, this is like a very complicated protein molecule formation. How this is formed, like you can start with that methionine is made, and then you can add this aspartic acid, that ASP, and then leucine, that is LEU, and triosine, that is tri. Now, each case what is happening, then when these two are added, then H2 molecule is released. Now, these two are added, then again H2 molecule is released, and that is possible because you have the OH and you have the H hydrogen. So, that forms that OH H2O. Again these two, when it is added, then OH group is released, H2O is released, and as a result these four comes together subunits, and then you can have again a C terminal and N terminal, and this is a polypeptide backbone structure, and you have the side chains that can have different side chains. It can have benzene rings, it can have methyl groups, you can have ethyl groups, all these ethyl groups. But that is remembering all this sequence, that how a complicated protein molecule forms is not important. What is important is the understanding overall how this protein structure can form. Like you have the different R1, R2, R3 group, they can come together, and every time one protein molecule, if this forms, then you have to understand that one H2O molecule has to be released. Okay. And if that H2O cannot be released, then protein molecule cannot be formed, simple. Because you need to ensure that your backbone chain is a polypeptide chain, and it has one end is a carbon end, one end is a nitrogen end. And typically according to the convention, your N terminal is always on the left hand side, your C terminal is always on the right hand side. It is just a convention. This is also schematic illustration of that bond rotation in amino acids. Now, this amino acid, the protein chains, also protein molecules, you can see that that is C alpha, that is one particular carbon atom orientation, and it is connected to another carbon atom, and it is connected to another nitrogen atom, and this is your essentially the peptide bonds. Now bond rotation means, now this is that individual amino acids. Now bond rotation means this bond angle they can rotate depending on what is the scenario, what is the situation, and if they can rotate, now this is one configuration. If they rotate to this side, then configuration also changes, you understand, and once it rotates, it can always come back to the original configuration also. So what I am trying to say here, this phi and psi angle, this phi and psi angle value is not a fixed value, they can dynamically change depending on the biological conditions that the protein molecules will experience in a particular biological environment. Okay. So overall, if I want to summarize that protein molecule structure, it has a polypeptide backbone chain, it can have different R1, R2, R3 different organic side groups, and these side groups can be either benzene ring based, or it can have a simple side groups like methyl or ethyl groups. Now then the other things that you have to remember, that when two different types of amino acids are coming together, they form a protein molecule, then that this addition of the two amino acids is only possible when they will able to release the water molecule. So that reaction must release water molecule, otherwise that polypeptide chain is never formed. The other things is that these bond angles between the carbogen connected to nitrogen and carbon bonds or carbon nitrogen bonds, that bond angle is not a fixed value, it can change during that any biological or pathological processes, and it can cause this change in the configuration of this protein molecule, and that protein configuration changes take place very dynamically during any biological environment. So I think I will stop here and we will start from this in the next